Hello everyone, welcome to today's session. Today we are going to see class 12 biology, chapter 5 molecular basis of inheritance, the DNA. So in the previous chapter we learned about inheritance, like how characters are passed from parents to offspring through Mendel's law. So Mendel called these units as factors. So today we know these factors are called as genes. But during Mendel's time, scientists they did not know what genes were made up of. So for almost 100 years, scientists worked to answer one important question. What is that question? So what is the actual heredity material inside our cells? But after many experiments, they discovered that DNA, that is the deoxyribonucleic acid, is a hereditary or genetic material in almost all living organisms. So just to think about it, see every characteristics, that is your eye color, hair color, height, blood group, even certain inherited diseases also, right, is controlled by the information stored in the DNA. So in class 11, you studied about nucleic acids. Yeah, there are two types, right? One is the DNA and another one is the RNA. So what are they? They are polymers made up of repeating units called nucleotides. Although both are nucleic acid, they perform different jobs, and we are going to see about DNA in this session. Yes, let's start with the topic, children. So structure of the polynucleotide chain. We know that DNA is a hereditary material, and let us understand how it is built. So DNA, it is an extremely long molecule. DNA is an extremely long molecule made up of repeating units called deoxyribonucleotides. Deoxyribonucleotides. Just imagine a long train. Each compartment of the train, it represents one nucleotide. So when millions or billions of these compartments join together, they form one enormous DNA molecule. And the length of the DNA is measured as the number of nucleotides or base pairs. Different organisms, they have different DNA length. For example, if we take this lambda bacteriophage, 48,502 base pairs are there, and Escherichia coli, 4.6 into 10 power 6 base pairs are there. So these are examples that show that the DNA can be incredibly long. What is a nucleotide then? Say a nucleotide has three parts. Just look at the figure. Instead of seeing all these things, I will explain you in this diagram itself, children. Just look at here. So this figure, it shows the basic structure of a DNA strand. So first component is the nitrogenous base. First component is the nitrogenous base. So these bases, they store the genetic information, and they are of two types. They are of two types. And first one is the purines. So purines, they have two rings. Purines, they have two rings. They are adenine and guanine. Adenine and guanine. You can able to see this orange color things, A, G, adenine and guanines. Next is pyrimidines. So pyrimidines, they have one ring, and they are cytosine, thymine, or uracil. So always remember, cytosine, this one, it occurs in both DNA and RNA. But this thymine, it occurs only in DNA. So instead of this thymine in RNA, it is replaced by uracil. Okay. So next component, we are going to see the pentose sugar. So now observe this pentagon shaped structures in the diagram, children. These things, yeah. So these represent the pentose sugar. So DNA contains deoxyribose sugar. So RNA contains ribose sugar. The only difference is that ribose has an additional OH group. It has an additional OH group at the 2 dash carbon. Okay, this is the 3 dash hydroxyl. So a 2 dash carbon, what happens? It has one OH group. So because of this extra hydroxyl group, RNA is less chemically stable than DNA. And the third component we are going to see is the phosphate group. So this is the phosphate group. So now look at these small circles here, joining one sugar to the next one. Yeah, this one, yes. So these represent the phosphate groups. So their main function is to connect the neighboring nucleotide. Is it connecting here? Yes. So it is connecting the neighboring nucleotides. And then we are going to see the formation of a nucleoside. The nitrogenous base first joins the 1' carbon of the sugar through an N-glycosidic bond. What happens? The nitrogenous base, it joins the 1' carbon of the sugar through an N-glycosidic bond, and this produces a nucleoside. Examples include like adenosine, guanosine, yes, cytidine or uridine. And if the sugar is deoxyribose, they become as a deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Yeah. Next, we are going to see formation of a nucleotide. So, nucleotide formation, a phosphate group joins at the 5' carbon of the sugar through a phosphoester bond. What is this bond? Phosphoester bond. And the now molecule becomes a nucleotide. Because of this, the molecule becomes a nucleotide. And then see this diagram again. Just notice how one sugar is connected to the phosphate of the next nucleoside. So these connections, these connections are called 3' 5'. That is a phosphodiester bonds are, you can able to see phosphodiester bonds, and these connections are called as 3' 5'. Yes, 3' 5'. And millions of bonds join nucleotides into one long polynucleotide chain. Next is the sugar-phosphate backbone. So just to see the outer portion of the diagram, children, you can able to see. So the alternating sugar and the phosphate molecules, they form the backbone of the DNA. And think of it as a side rails of a ladder. So it provides strength and support to the DNA molecule. And this nitrogenous bases, right? So they project inward. Yes. So they are, bases are not part of the backbone, but they extend inward like the steps of a ladder. They extend inward like a steps of a ladder. And these bases, they pair with another DNA strand to form the famous double helix. Let's understand this 5' 3' ends. See, just see the figure here. You can see the free phosphate group. Free phosphate group we can able to see. So this is the 5' end. And at the bottom, see this one at the end. Yeah, you can notice the free hydroxyl group attached to the sugar. Yes. So this is the 3' end. So every DNA strand, it has a direction. You can able to see here. Yes, it has a direction. Always it is a 5' to 3'. So this direction is called the polarity of the DNA strand. So this direction is called as the polarity of the DNA strand. And this becomes extremely important during the DNA replication and the protein synthesis, because new nucleotides can only be added to the 3' end. Yes. So let's see one quick exam tip for you. So RNA has an extra 2' OH on ribose and uracil in place of thymine. Yeah, we already saw these things. Let's move on to the next one. So here is discovery and double helix. We understood the basic building block of DNA, right? The next question is, who discovered DNA and how did scientists find its structure? See, this is one of the most interesting story in biology. So what happened in 1869? A Swiss scientist named Friedrich Miescher, Friedrich Miescher, he isolated a new acidic substance from the nuclei of white blood cells. What he did, he isolated a new acidic substance from the nuclei of white blood cells, WBC. Since it was found inside the nucleus, he called it as a nuclein. Yes. So at the time, nobody knew that this molecule carried the heredity information. For many years, what happened? Scientists knew that DNA existed, but could not determine its structure because DNA is extremely long and delicate molecule, and the technology available at the time was also not advanced enough to study it. And then what happened? After some time, a major breakthrough came when Rosalind Franklin and Morris Wilkins, what they did, they used X-ray diffraction to photograph DNA. What they did, they did X-ray diffraction to photograph DNA. So their X-ray images reveal that DNA has a regular helical arrangement. DNA has a regular helical arrangement. And using this X-ray diffraction data, James Watson and Francis Crick. James Watson and Francis Crick, they proposed the famous double helix model in which year? In 1953. And this model explained not only the structure of the DNA but also how this hereditary information could be copied from one generation to the next generation. And then another scientist, important scientist, Erwin Chargaff. Yeah, Erwin Chargaff. What he did, he studied the chemical composition of DNA from different organisms and he observed that amount of adenine is equal to amount of thymine and amount of guanine is equal to amount of cytosine. And this became the famous Chargaff's rule. Yeah. So these observations helped Watson and Crick to understand how the two DNA strands are held together. Let's see the diagram, children. So the diagram, it shows the two polynucleotide strands. This is the one strand and this is the another polynucleotide strands placed side by side. Notice, you can able to notice the sugar phosphate backbone. This is a sugar phosphate backbone lies on the outside of the both strands, right? So this provides strength and support to the DNA molecule. And the nitrogenous bases, it is projecting inward where they are pairing with the bases from the opposite strand. You can able to see, yes. And just observe this base pairs carefully. See, adenine, yes, it always pair with thymine using two hydrogen bonds. You can see the hydrogen bonds here, right? And then this guanine, it always pairs with the cytosine using three hydrogen bonds. So this pairing is called as complementary base pairing. This pairing is called as the complementary base pairing. So one strand, it is running from 5' to 3' and the opposite strand is running from 3' to 5'. So the strands run in opposite direction and they are called as anti-parallel strands. And another important point is that adenine and guanine are purines, right? So they have double ring bases. Whereas the thymine and cytosine, they are pyrimidines. So they have single ring bases. Always a purine pairs with the pyrimidine only. Why? See, this keeps the distance between the two DNA strands almost constant throughout the molecule. Two DNA strand almost constant throughout the molecule. Imagine just if two purines are paired together, they could occupy too much space, right? It will make the DNA wider. Yes. But if two pyrimidines are paired together, what will happen? There would be too much empty spaces. So it making the DNA narrower. So pairing of one purine with one pyrimidine, it maintains the uniform width of the double helix. What it does? Uniform width of the double helix, right? And then, why is this complementarity important? See, complementarity base pairing has huge biological significance. Suppose one DNA strand has a sequence. Just think, yeah, AT, GC, CA. Okay. So immediately we can predict the opposite strand, right? What you'll say? See, ATGCCA means easily you can say the opposite one will be TACGGT, right? So knowing the sequence of one strand, automatically we can tell the sequence of the other strand. So during DNA replication, each parental strand, it acts as a template. Each parental strand is that a parental strand, it acts as a template to produce a new complementary strand. So the daughter DNA molecules, it become identical to the parent DNA. So this how beautifully it is explaining the genetic information is passed accurately from one generation to the other. So next, We are going to see the base pairing and the helix dimensions. We know that base pairs, right? Let us understand the complete double helix structure proposed by Watson and Crick. So DNA, it consists of two long polynucleotide chain. You can able to see two long polynucleotide chain, right? And these two strands, they twist around each other, yes, to form a right-handed double helix. It forms a right-handed double helix. It resembles a twisted ladder. Yes, the sugar-phosphate backbone, it forms the outer side rails. You can able to see, yes, the outer side rails of the ladder, while the base pairs, it forms the rungs. You can able to see the base pairs in the center. So one important feature of the DNA is that it has two strands. Yeah, the two strands, they are anti-parallel. How they are? They are anti-parallel. So one strands run from 5' to 3' and another one runs from 3' to 5'. Let's see this diagram. Yes. So the entire structure is twisting like a spiral staircase. So the two outer ribbons you can able to see, and also it represents the sugar phosphate backbone, and between them are the base pairs which is holding the strands together, and each complete twist of the DNA is called as one turn of the helix. This one we called as the one turn of the helix. So in our NCERT book we have three important measurements we should know. So first one is the pitch of the helix. First one is the pitch of the helix or helix pitch. So what is this? The distance covered by one complete turn is called the pitch. The distance covered by one complete turn is called the pitch. The pitch of the DNA is 3.4 nanometers. Yes, nanometers. And now next one is the number of base pairs. How many base pairs are there? So each complete turn approximately it contains 10 base pairs. 10 base pairs. And next one is the distance between two adjacent base pairs. So this is one base pair and this is one base pair, right? Two adjacent base pairs. The distance is 0.34 nanometer. 0.34 nanometer. So these numerical values are very important for your board examination. Don't forget it. Yes, write and see all these things very important. Why is this DNA is stable? Yes. See, DNA, it remains stable because of two important reasons. First one is the hydrogen bonding. So what happens? Just remember, we know that AT, it has two hydrogen bonds, and GC, it has the three hydrogen bonds. So this GC has three hydrogen bonds. It is stronger than AT. And also it has base stacking. We now just observe how the base pairs are arranged one above another. Can you able to see how these base pairs are arranged one above another, right? So the flat base pairs stack like a pile of coins, right? How it is arranged. So this arrangement is called as the base stacking. And this base stacking, it provides additional stability to the DNA molecule. And DNA is stabilized by both this hydrogen bonding as well as base stacking. Next is the central dogma. So after understanding all this DNA structure, Francis Crick, he proposed the central dogma of molecular biology. What it states? It states that the genetic information flows in the following direction. How it is flowing? From DNA to RNA. From RNA, it is flowing to the proteins. So DNA to RNA, we call that process as the transcription. And from RNA to protein, we call that as the translation. So the proteins produced through this process, it determines the structure and function of every living organisms. So exception is the reverse transcription. So what happens in some viruses, example like HIV, they have RNA as the genetic material. So instead of copying DNA into RNA, these viruses first convert the RNA into DNA using enzymes reverse transcriptase. So this process we called as reverse transcription. Yes. So go through, don't forget, go through all this thing, children. This very important topic. Yeah. So next we are Packaging of the DNA helix. So far we have learned that DNA is a long double helical molecule. So now let us think about a very interesting question. How does such a long DNA molecule fit inside a tiny cell nucleus? See, the DNA present in a single human cell is about 2.2 meters long. How much? 2.2 meters long. But the nucleus has a diameter of only about 10 power minus 6 meter. That is 1 micrometer. Imagine trying to fit a 2.2-meter long thread inside a tiny ball. It seems impossible, right? But nature has an amazing solution called as the DNA packaging. Let's see a DNA packaging in prokaryotes. So prokaryote, they do not have a true nucleus. Their DNA is found in a region called the nucleoid. And the DNA, they are negatively charged because of the phosphate group. So to fit inside the bacterial cell, it forms large loops. We have already studied our bacterial cell, right? So it has large loops and is held together by proteins present in the nuclear region. But although the DNA is compact, it is not wrapped around the histone proteins like in eukaryotes. And DNA packaging in eukaryotes. Now let us move to the eukaryotic cells such as the plant cells and animal cells. See, this eukaryotic DNA is much longer. It requires a more organized method of packaging. It requires a more organized method of packaging. So nature uses the special proteins called histones. Histones. So histones are positively charged basic proteins because they are rich in the amino acids lysine and arginine. Why is this important? So remember that DNA is negatively charged due to its phosphate backbone. The positively charged histones attracts the negatively charged DNA, allowing it to wrap around them tightly. Histone octamer. Next we are going to see. Yeah, along with the picture I'll teach you. Just look this picture. So at the center you can able to see a group of 8 histone proteins. So this is the histone octamer. Yeah, you can able to see a group of 8 histone proteins. This group is called as the histone octamer. So it consists of 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4. So these 8 histones, they form a spool-like structure. And the next is nucleosome. So how a nucleosome is formed? Just observe the DNA strand. Yeah. So instead of remaining straight, the DNA, it wraps around the histone octamer. Approximately 200 base pairs of DNA, they coil around one histomer octamer, right? Histone octamer. So this complete structure is called as the nucleosome. This complete structure we called as the nucleosome. Yes, a nucleosome, this is the basic repeating unit of chromatin. And you can compare it to a thread like wound around a spool also. So this is the histone octamer. Yeah, this is a spool. And this is the DNA thread. Yeah. So just observe this diagram. So at the center, you can able to see the histone octamer. Around it, DNA is wrapped nearly 2 turns, and this one complete DNA histone unit we called as the nucleosome. And this is the smallest structural unit of chromatin. Next, we are going to see beads on string appearance. Yes, this one, this picture. So this is an electron microscope image. Just notice that this chromatin looks like beads connected by a string, right? So each bead is a nucleosome and the string is the linker DNA connecting the neighboring nucleosomes. So this appearance, we call this as the beads on a string model. So from chromatin to chromosome. So packaging does not stop here. So what happens? The nucleosome, it further coils and folds repeatedly. These coils, they become thicker and shorter. So during the cell division, they become highly condensed. And what it forms? It forms chromosomes. So how the sequence is now? DNA, it forms the nucleosome. This nucleosome, it forms the chromatin fiber. The chromatin fiber, it forms the chromosome. So let's see some exam tip. So euchromatin and heterochromatin. Euchromatin, they are loosely packed chromatin. Since it is loosely packed, enzymes can easily reach the DNA. So therefore, what happens? Genes present here are actively transcribed. Next one is the heterochromatin. They are tightly packed chromatin. Yeah, they are dense, right? Tightly packed chromatin. Because it is highly condensed, enzymes cannot easily access the DNA and they remain mostly inactive. Yes. Now let us quickly revise everything what we learned in this part. So DNA is the genetic material, yes, of almost all living organisms. It stores the heredity information and also controls the structure and functioning of every cell. So the basic unit of DNA is the nucleotide, which consists of the nitrogenous base, deoxyribose sugar, and phosphate group. When only the base and sugar are present, the molecule is called a nucleoside, and when a phosphate group is added, it becomes a nucleotide. So this individual nucleotides, they are joined by 3-5 phosphodiester bond to form a polynucleotide chain. So the sugar and the phosphate, it forms the backbone, while the nitrogenous bases, they project inward. And the DNA, it consists of two anti-parallel strands. So one strand runs from 5 dash to 3 dash and another one runs from 3 dash to 5 dash. So the complementary base pairings occurs, yeah. So and it follows the Chargaff's rule. So A pairs with T through 2 hydrogen bonds, G pairs with C through 3 hydrogen bonds, and the DNA molecule forms a right-handed double helix. So important dimensions you should remember. So pitch is 3.4 nanometer and 10 base pairs per turn, and distance between the adjacent base pairs is 0.34 nanometer, and the DNA, it remains stable because of the hydrogen bonding and base stacking. Yes. So in eukaryotes, what happens? DNA wraps around the histone octamers to form the nucleosome, and it gives giving the chromatin the characteristic beads on. What it does, the beads on string appearance. So these nucleosomes, they coil further to form the chromosomes. So just remember all these things, children. Don't forget. Watch the full video. Yes, we'll see you in the next video. Thank you.